Welcome! Today, we're diving into some really incredible research that tackles one of the biggest challenges in medicine. What happens when lung cancer spreads to the brain? You see, it builds this fortress that our body just can't seem to break down. But what if scientists just found the key to crack it wide open?
Right, so this is the central question, isn't it? The brain is supposed to be the most protected organ we have. So how can a tumor get in there and our own immune system, our best line of defense, suddenly becomes powerless? It's this massive puzzle that scientists have been working on for years, and figuring it out, well, that could change everything. So to get to the bottom of this, researchers came up with a really clever study. They took 30 patients, and for each one, they analyzed samples from the original lung tumor and from the new tumor that had spread to the brain. Think about it. By comparing these matched pairs, they could literally hunt for the clues, the exact changes that let the cancer not just survive, but actually thrive in that new environment.
Okay, let's get into what they found, because this is where we get our first huge clue. And it turns out the answer wasn't some single rogue gene. It was about the tumor's entire personality.
Now, to really get this, we have to talk about a concept called intratumor heterogeneity, or ITH for short. I know, it sounds super technical, but the idea is actually pretty simple. It's just a way to measure how different the cancer cells are from each other inside one tumor. Is it all one uniform thing, or is it this chaotic mix of all sorts of different cells? That one question turned out to be the key to the whole thing.
And here's what they found. It's a really striking difference. The original tumors in the lungs, they were like this disciplined army. All the cells were pretty similar, very uniform, with low genetic diversity. But the tumors that actually made it to the brain, complete opposite. They were like a chaotic rebel force, with tons of different cell types and super high genetic diversity. That chaos made them incredibly adaptable and unpredictable. And that right there, that was the big aha moment. This wasn't just a fluke, it was a pattern they saw again and again. The tumors that actually succeed in invading the brain are the ones with the most internal chaos, the highest ITH. So the next big question is, why? Why does all that diversity give them such a massive advantage? Well, that leads us right to the next piece of the puzzle. It turns out this diversity gives the cancer not one, but two totally different ways to hide from our immune system. It's like the tumor has two secret playbooks it can use to survive. So check this out. The researchers figured out that these tumors have two completely different survival strategies. The high-diversity tumors are rebel force. They tend to just go invisible. They literally get rid of the identification flags, the antigens that our immune cells look for. This creates what's called a cold tumor that just flies right under the radar. But the low-diversity tumors, they use a different trick. They put up a big stop sign using a protein called PD-L1. The immune cells show up, they see the tumor, but that sign basically tells them, nope, nothing to see here, move along.
And honestly, this is the real breakthrough here. For so long, the thinking was that all brain tumors were basically using the same old tricks to hide. But this research, it shows that's not true at all. It's a parallel system. They're choosing one of two very different paths to stay alive. And knowing which path they've taken, well, that's everything.
So what does all this mean for an actual patient? I mean, this isn't just some interesting fact for a science textbook. This choice of hiding strategy, it has a direct and really profound impact on a person's chances of survival.
Yeah, this is where the science hits the real world. That little strategic choice the tumor makes, whether to throw on an invisibility cloak or to just put up a stop sign, it turns out to be one of the most critical factors in determining how a patient will do.
I mean, just look at this chart. The data is absolutely stark. Patients whose brain tumors had high diversity, the ones that go invisible, they had a significantly worse prognosis, a lower survival rate. But patients with the low diversity tumors, the ones using the stop sign, they had a much, much better survival rate. It just goes to show you how critical it is to know exactly what kind of enemy you're up against. But, and this is the most hopeful part of this whole story, now that we understand these two different strategies, we can write a brand new therapeutic playbook to fight back. So instead of trying the same treatment on everyone and just hoping it works, we can finally move toward truly personalized medicine. The idea is simple, but it's so powerful. First, you figure out the tumor's hiding strategy.
And then you pick the exact weapon designed to beat that specific trick. So here's how the new playbook would work. Step 1, you profile the tumor to see its diversity level. If it's a high-diversity invisible tumor, you hit it where it's weak. Its chaotic nature makes it vulnerable to drugs called PARP inhibitors that attack its DNA repair system. But if it's a low-diversity tumor, using that stop sign, you use a totally different weapon. Immunotherapy drugs that block that sign, effectively taking the brakes off the immune system and letting it do its job. Now, of course, this is just the beginning. The road ahead is pretty clear. Scientists need to run bigger trials to confirm all this and then start actually testing these personalized treatments in patients. They're also looking to see if this same pattern holds true for other types of cancer. And what's really exciting is they're exploring tools like liquid biopsies, which could potentially track a tumor's strategy with just a simple blood test. Imagine that. And that leaves us with this final, really powerful question. This research, it's more than just a new way to fight brain metastases. It gives us a whole new principle. What if the real key to personalized cancer care isn't just about finding one single broken gene, but about understanding the tumor's overall complexity, its diversity? Profiling a tumor's personality might just be the future of how we treat cancer.